Genetic cloning. The past knowledge I had before researching was that cloning is a very controversial topic. It is still developing. It will change the world forever. You're not allowed to clone humans. It involves a lot of DNA analysis, and majority of people and a lot of people without the common knowledge believe it is unethical. And I also knew that the first mammal to be cloned was Dolly the sheep. I personally chose to research about cloning because it really piques my interest because of all the stigma and negativity around it. And I wanted to know more about the technological and medical advances that can be created, such as organ cloning. The big question is, what is genetic cloning? Well, cloning is the creation of a copy of a cell or of an entire living thing. So essentially, a clone has exactly the same DNA as the original cell or living organism. So there are many different types of cloning, and the main three types are reproductive, therapeutic, and DNA or gene cloning. Genetic cloning is when biological parts of an organism are reproduced and used for fields in medicine and agriculture. There is also therapeutic cloning, which is when you swap nucleus DNA between eggs for a shorter growth process. The most known is somatic cell nuclear transfer, which is a big deal in the cloning world because Dolly the sheep and several other successful clones occurred from this technique. Reproductive cloning is defined as the deliberate production of genetically identical individuals. Each newly produced individual is a clone of the original. And monozygotic, which is identical twins who are natural clones, and cells from two clones have the same DNA and the same genes in their nuclei. Who discovered genetic cloning? In the late 1800s, Hans Driesch was the first man to clone animals. He split a sea urchin embryo, and Hans Spemann also did a similar experiment with a salamander in 1902. In 1903, plant physiologist Herbert J. Weber coined the term clone from the Greek klon to refer to the technique of propagating new plants using cuttings, bulbs, or buds. He was the first to use the word clone for plants, but it later got changed to all organisms with identical DNA. How does genetic cloning work? These are the steps used in genetic cloning by researchers and scientists. Step 1. Extract DNA from a donor. First, scientists take healthy, viable cells from the organism they are trying to clone. They usually aim for somatic cells, which are the neutral type of cells that does its job and has two sets of chromosomes. Somatic cells aren't found in red blood cells. They are only found in white blood cells and are a common source for DNA products. The cells have to be healthy and not damaged. This is why it is very irrational for researchers to try to clone ancient frozen or trapped animals because their cells are mostly damaged. Step 2. Prepare an egg cell. While some scientists work on extracting somatic cells from the organism, others try to prepare a viable egg cell. It doesn't have to be an egg cell from the same species, but for better success rates, that would be ideal. When scientists find the right undamaged egg cells, they carefully extract the nucleus of the cell. The nucleus is what holds the single set of chromosomes that contributes to reproduction. But for cloning, they don't want that DNA. They want an intact, empty shell that can house an embryo. This means that the nucleus and all its DNA is removed, while the rest of the egg is delicately preserved. Step 3. Insert somatic cell material. Somatic cells are complete and adult cells are not used in reproduction. They have two sets of chromosomes, but scientists want to get this DNA into the egg cell and make it grow into a new organism. They have to remove the nucleus, insert it into the empty egg cell, and wait. Their goal is to combine them into a single cell again, which is very hard to achieve. New successful techniques use a light-directed flow of energy, causing the nucleus and egg cell to join and stay together. Step 4. Convince the egg that it's fertilized and implanted. So after that egg is cloned, it is ready to start growing. The egg does have two sets of chromosomes, but it needs to grow into a copy of the donor organism. In reality, the egg has not been fertilized and can't be without the cloning process being ruined. After the cell starts to divide, scientists have to move quickly and keep the egg in similar conditions to the real reproductive processes that occur naturally. If the egg starts to develop into an embryo that's healthy, they implant that into a living female organism to gestate. This is a better option for the egg and inexpensive compared to growing an embryo outside in a lab. Step 5. Repeat until viable. There is a lot of uncertainty in this process. Even the tiniest amount of cell damage can ruin the cloning process. There's no guarantee a healthy egg cell will develop properly internally or externally. Viability of the egg is a major concern. There are numerous failed attempts and embryos that don't develop correctly. It takes immense time, resources, and hundreds of attempts to create a successful clone. Successful live births are extremely rare as well. The process is extremely difficult for the clones as well. They have been known to suffer from short lifespans and have problems with their DNA. Most of these problems have been solved in recent years, but there is still a lot of research that needs to be done. How and where is cloning applied? Well, cloning in animals has been used in many different fields of study. For humans to have better lives and have a more efficient source of living. Animals have been cloned to have gene mutations that can assist scientists in studying diseases that occur in certain species. Livestock like cows and pigs have also been cloned to produce more milk or meat for human consumption and mass production. There is a lot of unethical processes and treatment of animals that are an issue as well, but most ethical research and scientists that are conducting these experiments have to follow the ethical protocols. The medical field. In the medical field, human cloning has been used to make early embryos, which is a revolutionary step for medicine. The cloned embryos were used as a source for stem cells, which can make new heart, muscle, bone, brain tissue, or any type of cell in the body. This is extremely useful in humans who have organ failures, cancers in their cells, or many diseases that can hopefully be cured in the future with these technological advances and processes. Research and science purposes. Cloning is also obviously used in science. Researchers can use clones in many variety of ways. They can also turn them into nerve cells to fix damaged spinal cords or insulin-making cells to treat diabetes. There is a definite pattern of making lives easier and more efficient for humans and finding ways to sustain living longer and for diseases and many organ failures to be cured. In the agricultural world, clones allow farmers to upgrade the quality of their herds by providing more copies of the most efficient animals, which in turn make more of those animals, and the animals are then used for conventional breeding, and they naturally reproduce the offspring, and they become the food producing animals. Scientists use it for research purposes, and in the future, it may be used for agricultural purposes, but it is still in the research stages for now. Plant cloning is a huge part of agriculture. Seeds genetically identical to parent plants could revolutionize agriculture because the production of the exact genetic replicas of important food crops can come a step closer because of the ability to clone the best crops. This could revolutionize our world because with biotechnology and genetic engineering and breeding plants, we can take all the defectiveness out of our crops and yield them to stand temperatures and the climate. Historical development. Is it recent and has it changed over time? What historical and current examples exist? Cloning has been around for a very long time. It was discovered in the late 1800s and has been redeveloping and enhancing based on the new technologies and advances being discovered. It has very much changed over time, and the timelines below explain in detail the different advances being done in cloning. In 1952, Robert Briggs and Thomas King describe how they cloned frogs by replacing the nuclei of eggs with cells from tadpoles and adult intestinal epithelium. 1984, Chinese researchers cloned a fish from cultured kidney cells. Researchers in Scotland cloned two lambs from embryonic cells. This was a crucial step towards cloning an animal from an adult cell and is seen by some scientists as a bigger breakthrough than Dolly. Roslin researchers then announced the birth of Dolly, who was the first mammal to be cloned from an adult cell. This ignited public debate about the prospects for human cloning. 1998, scientists in Hawaii revealed the cloning of 3 generations of mice from the nuclei of adult cells. This suggested that the technique could work on other mammals. Also, Japanese researchers reported cloning 8 calves using adult cells, raising the possibility that animals could be cloned for the quality of their meat and mass production. In New Zealand, scientists announced Elsie, who was a clone created from an adult cell from the last surviving Enderby Island cow. This proved that attempts to clone endangered species is possible, and it has been met with criticism because of the technique. Year 2000, Scotland Therapeutics unveils a litter of 5 cloned piglets. The firm said that genetically engineered cloned pigs could one day provide a source of organ transplants for humans. 2002 was the year the first cloned cat came to existence. It was announced by Texas researchers, and the cat's coat pattern is not the same as her genetic donors, showing the impact on development of non-genetic effects. 2003, Italian scientists announced the first horse clone created from a skin cell. In that same year, French and Chinese scientists unveiled Ralph the cloned lab rat. Rats were tough to clone because the eggs divided before the donor DNA is injected, so the technique relied on using drugs to create division. 2004, a researcher in Korea claimed to have derived stem cell lines from cloned human embryos, but was later discredited. This was probably the highest number of cloned human embryos, but there is no evidence for it. 2005, in the same researcher's lab, the creation of Snuppy the dog was confirmed. Although the stem cell research had been discredited from the lab, Snuppy's clonal credentials have been confirmed. 2007, reprogrammed stem cells were created, and they were cells that can potentially make any cells in the body. 2007, 4 gray wolves were cloned using cells from a dead wolf and putting the embryos in domestic dogs. That same year, the first cloned embryonic stem cells from primates were created by replacing the nucleus of an egg with the nucleus of an adult cell. 2010, the first fighting bull was cloned. In 2011, the first reprogrammed stem cells from the endangered white rhino was reported. 2011 was a year of breakthrough for cloning, when a transgenic cow produced by cloning with human genes was created with milk similar to human breast milk. This was also the year that human stem cells were cloned. Genetic material from adult skin cells were grown into an early embryo inside a human egg. Now, projects in Japan, Russia, and South Korea are all vying to resurrect the long-extinct woolly mammoth. There's no specific evidence that anyone has cloned human embryos. In 1998, scientists in South Korea claimed to have successfully cloned a human embryo, but said the experiment was interrupted when the clone was just a group of 4 cells. This is why they have been discredited and there is no evidence. Why is Dolly the sheep so important? Well, Dolly was the first mammal to be cloned from an adult cell. Her birth proved that specialized cells could be used to create an exact copy of the animal they come from. DNA tools and processes are used in cloning? Well, there are 7 important tools of genetic engineering. There's polymerase chain reaction, restriction enzymes, gel electrophoresis, DNA ligase, plasmids, transformation and transduction, and also identifying transgenic organisms. We learned in this unit, polymerase chain reaction copies a section of the DNA, and restriction enzymes cut the DNA strands into smaller pieces. The risk factors of genetic alteration. Scientists have found adverse health effects in sheep and many other cloned animals. These include an increase in birth size and a variety of defects in vital organs, such as the liver, brain, and heart. Other consequences include premature aging and problems with the immune system. How have societal pressures such as food, energy, and resource shortages driven the development of my topic? Cloning is a process that requires millions of dollars in research. It is an extremely limited process because of the funding needed, and it cannot always be regularly done. And not everyone can pursue the science of cloning. It takes very skilled researchers and scientists who have to train for years and gain a lot of background knowledge. There are a lot of resource shortages when funding is not available, and it is also very competitive. Scientists all around the world are competing for the new best innovation and way to make millions and revolutionize another section of science using cloning. Ethical concerns. There are many ethical concerns related to my topic because cloning has many different aspects to it and not everyone takes that into account. There are many ethical concerns regarding human cloning as well as animal cloning. People have a hard time wanting to accept the need for clones and how they are revolutionizing our world. A part of a controversial method of cloning is somatic cell nuclear transfer. It involves the production of a 2 to 4 day old blastocyst. The cells are removed to make a line of embryonic stem cells, a process that destroys the embryo. This brings the concern of killing an unborn child. Another concern is over what might be done with these embryos before getting a stem cell line. Misconceptions and misunderstandings of cloning. When people think of cloning, they immediately have a negative perception because they think of cloning humans and the unethical concerns that arise. They forget that cloning is used in agriculture and other aspects to improve human life. There is also a common misconception that cloning is a new technology, which it is certainly not. It has just been advancing and being enhanced because of the new technologies. Humans consume from plant clones regularly. Most of the non-organic produce sold at grocery stores are from cloning plants. Plant cloning is referred to as vegetative propagation. It takes about 30 years to breed a banana from the seed, so to speed the process of getting the fruit to the market, most bananas, potatoes, apples, grapes, pears, and peaches are from clones. Accessibility with the public. Cloning is not highly accessible to the public. Special genetic researchers and scientists that are approved to attempt this genetic change are allowed to. Human cloning is also strictly prohibited under the law. Under the AHR Act, it is illegal to knowingly create a human clone, regardless of the purpose, including therapeutic and reproductive cloning. Media's role in crafting public perception related to cloning is very deceptive. The reality of it is much different. There are many apocalyptic visions of cloning, and because of the media and perceptions of cloning related to aliens and the end of the world in movies, there are many negative connotations without knowing the actual effects, and people portraying cloning in a certain way will dictate how many people view new technologies and if they will fear it or not. Effects of cloning on society. Most scientists believe that the process of cloning humans will result in an even higher failure rate than today. Not only does the cloning process have a very low success rate, but the viable clone suffers increased risk of serious genetic malformation, cancer, and shortened lifespans. Society is scared of future human cloning and the evil that can occur if human genetics can be altered. Society would be split and everything in our world would change. In our current society, cloning is helping a lot in agricultural and medical fields and is a great advancement for us. I learned the effects of cloning and the big perception of it that media induces on everyone. I also learned how exactly cloning works and the biological processes of it. I learned how important it is for stem cell research and how it is positively impacting our world. I learned that there is also fear of cloning being used for wrongdoings. How it relates to me. It relates to my interests but doesn't relate to me personally. It relates to me in the sense that I eat produce that have been cloned through vegetative propagation. It also relates to me because the future of cloning will affect my future. If human clones are created, life will drastically change, and this will affect everyone on the planet. I'd like to find out more about organ cloning and the medical advances that cloning will enhance in the future. I would also like to know more about plant propagation and how we eat produce that are a product of cloning. Thanks for watching.